Alright, so today's Lab 12, just one experiment, bacterial transformation. So we're going to transform bacteria. We're going to use that using the pGlo system, right, which is made by the manufacturer Bio-Rad. So this is their design. So examples of transformation. So transformation, even though we're going to do it in bacteria, has been and continues to be done even for eukaryotic organisms, right? So these mice, when you take a look at this picture, you can notice that 2 of them are expressing green fluorescent protein. The third one is not. Why would you want, right? Granted, it was fun for me to draw on my Petri dish, right? But why would you want something to fluoresce green? So you can see it, right? The great thing about fluorescent proteins is you can see them, right? You can distinguish them from other proteins because they fluoresce. So a lot of times it's used as a marker. In other words, to allow us to visualize a genetic change, because this is genetic change, y'all, that we're going to cause to happen today. We're going to actually put DNA into the bacteria that allows them to make this protein. And that's a very important point, right? Transformation is actually adding DNA that the organism can utilize to then transform, right? To do something different. So my favorite one, and kind of gives you an idea of how beneficial fluorescence could be, is this fruit fly sperm video. And whenever you say sperm, usually people pay attention to you too. And of course, it's not going to my video. Why isn't it? Open hyperlink. So you can see here, this is, here I'll blow it up. This is the reproductive tract of a female fruit fly. And what you see swimming around are, of course, the sperm that she's been inseminated with, or mated with. Yeah, so this is done probably with magnification, with a microscope or a lens of some sort. Yeah. The really cool thing is if you look closely, you really see the green guys, right? The green sperm swimming around. But now look, and you can see the red ones now too, right? So what they did is they made one male fruit fly, his sperm, they made them red. And the other male fruit fly, they made his sperm green. And female fruit flies will mate with more than one male. And they'll do this so that hopefully their offspring are diverse, right? And therefore there's a more likelihood that they'll survive in the environment, right? Under the pressure of the environment. Survival of the fittest, right? So if you mate with more than one male, the chances are at least offspring from one of those males will be good. Make sense? So what they found is that the sperm themselves actually compete to get to the egg. Right? And they were able to see this, that the different sperm are able to recognize each other by the fact that they made them different colors. So they were able to watch the behavior. And then they were able to go further too, because obviously the female, the reason why she mated with multiple males was she wants diverse offspring. So if the sperm are fighting to get to the egg and maybe only one of them wins, one of their males wins, that wouldn't be beneficial to her, right? Why even mate with multiple males? So she actually produces substances in her reproductive tract that inhibit competition between the sperm, or lessen the competition. Kind of interesting, right? None of this would have been made possible if they weren't able to genetically alter the sperm so that they could see the difference between one male sperm and another male sperm. Right? Scientists think of these things. No, not really. You're trying to ask a question.
it like when they're integrated to the gene? Is it like a transposon mechanism?
Oh, we're going to talk about it. We're going to talk about it. Getting ahead of yourself. That's okay. Okay. We're going to do it in bacteria, and this is in bacteria, right? So this is bacteria growing on a petri dish, and they just use different ones that are fluorescing different proteins, right? So green fluorescent protein and red aren't the only ones. There's a whole bunch of other fluorescent proteins out there, and so somebody had a lot of fun, right, making this particular one. We've done this with the kids in the past in the summer. We just didn't have as many colors for them to choose from, where they've drawn like I did. But they drew on really huge petri dishes and worked with the art department, so it was kind of fun. So that's for fun, right? This is real science. So what is bacterial transformation? It's where DNA comes from a source. And in this case, unlike in nature, we're going to be starting with a pre-made circular piece of DNA known as a plasmid. In nature, what happens is typically bacteria will die and their chromosome, their DNA, becomes part of the environment. This can be picked up by other bacteria and incorporated into their chromosome. If they incorporate it and utilize it, they're said to be transformed. And I always think of transformers, right? You can go from a robot to a car, right? You can make that big transformation. So just as you see here, DNA is that information. And it can be used to make anything. In this case, it's going to be a fluorescent protein. So a transformed cell has taken up a portion of DNA from another organism and integrated it into its own chromosome. So why is this important? Why do we talk about transformation? Why do we do transformation? Well, unfortunately, some organisms, that information they picked up allowed them to become what we call superbugs. Why do we call them superbugs? What abilities do they have? What makes them super? Anyone know? They can protect themselves. What do we usually use to kill bacteria? We use antibiotics to kill bacteria. Can we use any antibiotic we want on any bacteria? No. Some bacteria have become what we refer to as resistant, right? And one of the ways that they've acquired this resistance is picking up DNA from other resistant bacteria that infers that ability, right? So remember, where do antibiotics have to go in order to affect a cell? They got to go inside the cell. And they got to bind to some molecule, usually an enzyme, maybe even the ribosome, right, and interfere with some process that's crucial to that cell. So we talked about resistance, we talked about antibiotics before, right? So what's some ways that bacteria are resistant to antibiotics? How are they resistant? Too much exposure, but at a molecular level. So antibiotic comes in. Why doesn't it work sometimes? What does it have to do in order to work? What did I say it does? What, Alerica? It has to get into the cell to begin with, right? So some of them have resistance in that they've stopped the uptake, right? It doesn't even get into the cell. So in the case of gram-negatives, that outer membrane sometimes is a really good antimicrobial or antibiotic membrane, and that it's hard to penetrate. So E. coli, remember, penicillin can't even get in. Okay? So that's one way, right? Stop it from getting in. Or once it gets in, do what? Once an antibiotic gets in, it's got to bind to something, right? So what if what it normally binds to isn't there anymore, or it's changed so it can't bind to it? Can it bind? No. Some can kick it out, right? So it comes in and they're like, oh, no, that's bad news, and throws it back out. Right? Others, when it comes in, they destroy it with enzymes, right? Which is the case for a lot of organisms now, they have enzymes that digest the antibiotic, right? So antibiotic comes in, enzyme breaks it up, destroys it. We're going to use one of those enzymes today. How does the cell know how to make that enzyme? Where is that information encoded? In their DNA, right? So in their DNA, they have the ability, the information to make this enzyme that destroys penicillin. So we discovered, this was discovered in 2009. A Swedish man acquired a Klebsiella pneumoniae infection in India, and it was resistant to a particular class of antibiotics. And the reason why it was resistant, right, so here is the drug. It's very similar to penicillin. It's in that same grouping. So it has this ring structure right here, what is referred to as a beta-lactam ring, right? Ampicillin has it, penicillin has it, a lot of the psyllins as well as this other drug have it. I don't even know how to say that one. I need to figure. Carbapinin? I have no idea. It's a tongue twister. So what they found out is that this Klebsiella pneumoniae strain that he acquired contained a plasmid, so an extra circle of DNA, right, that some bacteria will have, right? So it has an extra DNA other than even its chromosome. On this DNA circle, this plasmid, they found the gene that they named BLA, and then the abbreviation New Delhi for where he acquired it. Melito, and then beta-lactamase is the BLA part, and that stands for the name of the enzyme. The enzyme that digests this beta-lactam ring structure right here is referred to as beta-lactamase, right? So they found the gene, the DNA, that told that bacteria how to make this enzyme. So when that antibiotic goes in, enzyme digests it and stops it from working. Still with me? Okay. So similar strains of Klebsiella pneumoniae contain enzymes. This gene was transferred horizontally to other species of bacteria. So ways in which bacteria can move DNA from one bacteria to another. So those of you guys that are in my lecture class, you know transformation is one of the ways. Another way is the bacteria literally join up by the sex papillae. What process is that? Conjugation. And then the other way is if a virus picks it up and transmits it to the other one. That's the other way of lateral gene transfer, and that one's called transduction. Oh, good. You guys were paying attention. Final exam's going to be a walk in the park, right? And then transformation, right, which is what we're going to do today, where they just pick it up from the environment, that DNA. So this can happen any one of those ways, right? DNA can laterally move from one bacteria to another. Which specific mechanism, whether it was conjugation or transduction or transformation, we don't know. We don't know, right? But we do know it moved laterally. As of October 2015, there was a higher than expected level of resistance in Enterobacterase to this antibiotic. In a report that was put out by Atlanta, Baltimore, and New York hospitals, right? And this is where we use most of our antibiotics, right, is in hospitals. And now we're finding the more and more bacteria present that are resistant to the antibiotics we're using to treat our patients. So by August 2010, isolates that contain this gene, right, this ability to make this enzyme, and to multiple different classes of antibiotics, right? So this is only one type of antibiotic, but there are other antibiotics, aminoglycosides, fluoroquinones, But they were still susceptible to polymyxin, antibiotic, and colistin, right? We used colistin in our blood agar plates. Although colistin is highly toxic to the human kidney cells, and therefore it's difficult to administer. We can use it safely, right, in our plates, but you wouldn't necessarily want to just, prescribe willy-nilly to your patients. Not that antibiotics should be used that way. It's part of the reason why we're experiencing the problem we have today. So most of us know about MRSA, right? Mictillin-resistant Staphylococcus aureus, right? Where Staph aureus strains have become resistant to that very common penicillin derivative that we use. And we've got extremely drug-resistant strains of tuberculosis as well, where there's very few drugs left to give. So if they become resistant to those drugs, can we cure someone with that type of tuberculosis? No, and that's the scary reality of the world that we live in right now. This isn't the future, y'all. This is now. We're running out of antibiotics, right? Because of misuse and the natural ability of bacteria to change and acquire DNA from one another and acquire these abilities. So those of you guys in my lecture class, I already talked about this one, right? E. coli 0157. So this is a pathogenic strain of E. coli. And the pathogenic ability of this one is that it produces the Shiga toxin. This is a highly damaging toxin to your intestines. That's why if you have this strain, you wouldn't be in class right now. You'd be running to the bathroom. And it would not be pretty. The Shiga toxin is named after Shigella. It's a bacteria similar relative to like Salmonella. And why it's such a nasty infection, again, is because of this toxin. So how is it that E. coli, this strain of E. coli, has that toxin? How does it know how to make a toxin? Where's that information in the cell? In its DNA. So for the Shigella, how does it know how to make the toxin? It's in its DNA. So what does that tell us? E. coli got DNA from Shigella. The question is how, right? Lateral gene transfer is how. Which method? We don't know, right? Was it transduction? Was it transformation? Was it conjugation? We don't know. We might never know. The fact is, though, is it got it, right? This information moved from one bacteria to another. Does that make sense to you guys? Right. So transformation can be used for lots of things. When I was in research, E. coli are really cheap machines. They copy DNA really well. And so scientists like myself would use them to even just make copies of DNA, right? We would transform them. We would give them DNA and get them to copy it for us. And then we would just lyse the cells and take the DNA from them that they copied for us. But there are, other ways that scientists utilize this stuff. So one of my favorite transformations that's been done is they've taken the information on how to make human insulin, right? So how we make that protein. And they actually put that into bacteria. And so bacteria make, in the laboratory for us, insulin. But biotechnologists didn't just stop there. They've actually tweaked that protein structure so that now the insulin that it produces is more stable at room temperature. And people who are diabetics don't have to carry around refrigerated insulin, right? They can keep it at room temperature and it doesn't denature that protein. It's still effective. So it's really cool, right? Beneficial things that we can do with this type of science. So the possibilities are truly endless. But when it comes to modifying our foods, I have to say that I'm not on that bandwagon. There's not enough. We don't know enough. I don't feel we know enough. Yeah. So, natural competence. About 1% of known bacteria are naturally what we refer to as competent. So in other words, have this ability to take up DNA from the environment. Transformation in nature, right, is actually quite complex. For example, Bacillus subtilis takes about 40 different genes to acquire and have this ability, right? So not all bacteria have this ability, not all bacteria do this, but quite a few do. It does require energy to take up that DNA from the environment. It's not free. We believe it's evolved in a way to repair damaged DNA. So for bacteria, they're haploid. Typically, they only have one copy of all their genetic information. So if their DNA gets damaged, right, one of the ways to repair it would be pick up information from the environment. Hopefully, you know, it's somebody closely related or nearby you, and you can patch your breaks and your damage with the correct information. It's sometimes referred to as a primitive way of sexual reproduction. So especially with conjugation, right, sometimes I refer to it as bacterial sex, right? Because that's what sex is. It's exchange of genetic information, right? But it's not required for bacteria. Right? They can reproduce asexually. They don't have to exchange genetic information. So we cause bacteria to be artificially competent when we do transformation in a lab. There are two main ways that scientists use to make them competent so that they will take up the DNA and therefore be able to be transformed. You can do electroporation, and as the name implies, right, we use electricity to force the DNA into the cells, right, makes the membranes more permeable by hitting it with the electricity, and therefore the DNA is able to be taken up. And we can get it to take up other things, right, not just DNA. They can do it to get it to take in chemicals or drugs or any number of things. And this can be applied to eukaryotic cells as well as prokaryotic cells. Right? We can use this technique to get DNA into other types of cells, not just prokaryotes. Obviously, we don't do this here at Delgado because this type of technique requires special equipment, right? You need a machine to apply the voltage. You need special chambers in order for the reactions to happen. But I do want to make you guys aware of it, right? That in some research laboratories, this is how they do competency. Another way is chemically treated cells, and that's what we're going to employ today. Chemically, we're going to use a solution that's referred to as a transformation solution. It's a calcium chloride solution. And these ions, in the concentration that we use, affect the membrane permeability. And so therefore it's going to allow us to get the DNA into the cells by making the membranes of these cells more permeable. We're also using a special line of E. coli. It's not your straight up plain old E. coli. We're using a special line of E. coli that is more susceptible to competency chemically. So the calcium ions, remember, membranes, DNA, these types of things are highly negatively charged. So the positive ions of the calcium help shield and those negative ions, and allows for this to happen, for the DNA to be able to get in. The DNA is actually only taken in a single strand. So even though we have double-stranded DNA, only a single strand is actually going to enter into the cells during transformation. For this experiment, we're also utilizing a plasmid, right? So a circular piece of DNA. And in this case, the whole thing will go in. So there's commercially available chemically competent E. coli kits that a lot of laboratories, especially the one that I worked in, utilize. They have different efficiency rates, right? How good are they at picking up the DNA, right? And obviously, depending on what type of research you're doing would determine which efficiency rate you need, right? How many transformed cells do you need to be able to recover from this transformation? So it comes with all the supplies that you need, and even a controlled plasmid, right? To make sure that your system is working. So what's transformation? It's a process by which bacteria cells take up, if we're talking about bacterial transformation, exogenous DNA molecules, so outside the cell, and bring it in. Competent means the ability of the cell to be able to take up the DNA, right? Not all cells are competent. Bioluminescence, right? So this is the product or the emission of light by a living organism as the result of a chemical reaction during which chemical energy is converted into light energy. And so, anyone know where we discovered fluorescent proteins originally? Jellyfish. Yep, from jellyfish. So if you go to the Aquarium of the Americas, one of my favorite places to go is the jellyfish exhibit. That's why they're kind of in dark chambers too, is so that you can help visualize them. There is one that's one of my favorite ones. It has these little tiny cilia-like things on them, and they beat back and forth. But if you read the little plaque, and it looks like little rainbows, right, running along them. But if you read the little plaque next to it, they're not fluorescing. What it is those little tiny projections that are like cilia are like little tiny prisms, and they're refracting the light. You know, just like a prism reflects the light, separates it into the different colors, and you see a rainbow. The same thing is happening for that particular jellyfish, right? So that's a refraction of light that's happening due to their structures. I don't know if they have any fluorescent ones. I haven't had a chance to ask my friend that works there, and I haven't been in quite some time, unfortunately. But definitely, you know, read the plaques, right? And who knows, maybe they do have it there, right? And you could find them and see that fluorescence. But it's very common in jellyfish, but not all of them have this ability. So what happens is that when we shine the UV light on it, it's absorbed and electrons are moved. And when those electrons move back to their ground state, they're excited and they emit the color, right? So we can say it in technical terms, right? But in general terms, that's what happens. And it's at a different level of energy, right? Not as much energy is given back. So the take-home message here is that smaller wavelengths have higher energy. So UV light has a really small wavelength, so much so that we don't see it. We don't actually see UV light. But when it fluoresces back, right, that's within our visible light spectrum. So notice UV lights, as I said, really small, about a nanometer in wavelength, where visible light, right, are much larger wavelengths, 500, 700 nanometers, right? And that's why we're able to see them. So all the conditions have been worked out for today, right? It is a system, it is a kit, right? We don't have to work out any of the details. We know how many cells to use. We know how much DNA to use. We know the transformation efficiency of this system, how many transformed cells we should get per milligram of DNA that we're going to add to our samples. We're going to spread a set amount, 100 microliters, onto our plates. And we should get around 75 transformed cells per plate if everything goes well. Normally you'd have to do serial dilutions, right? Similar to what I demonstrated before, and actually calculate out your colony forming units to determine efficiency. So this is the plasmid that we're utilizing. So again, circular piece of DNA, and it contains genetic information. It contains information on how to make things. So for this experiment, we're actually going to give the bacteria beta-galactonase, ampicillin resistance. Right, so we're going to give it that enzyme that we talked about earlier that allows it to break the beta-lactam ring. So in this case, we're using ampicillin, which is a penicillin derivative, so it has that beta-lactam ring. So these organisms will make this enzyme and have the ability to destroy that antibiotic. Right? They'll become ampicillin resistant. Another reason why we're definitely not taking them out of the lab, right? Everything in lab stays in lab. The other thing we're going to give it is the genetic information on how to make green fluorescent protein. Right? So in the DNA, in this code, this is how you make green fluorescent protein. And as I said, this is originally derived from jellyfish. We're also going to give it the ability to make this protein coded ARAC. It's a regulatory protein. And then all plasmids have a site of origin for replication. So for the organism to be able to copy this circular piece of DNA, this plasmid. And that's the site of replication where it starts. This gene, ARAC, this regulatory protein, is what's referred to as constitutively expressed, which means that all the time, right, it's making this protein. Do we always make everything we have the ability to make in our DNA? No, right? Some of the stuff is said to be under control, right? And this protein is actually a control protein for a system of control that's set up in this plasmid. And it was actually borrowed from a system for digesting a sugar. It takes energy to utilize this information, so sometimes if it's not beneficial for an organism, they'll get rid of the information. So we're putting pressure on these bacteria in that we're like, you have to take this, you have to utilize it, otherwise you're going to die because we're going to put ampicillin in front of you. And so if you don't keep this, you don't make your ampicillin resistant enzyme, you're going to die. Right? So we put that selective pressure on them. That's why one of the reasons why we use the antibiotic, why we give them antibiotic resistance. Right? So some terms that go along with this system, right, of controlling our DNA. Operons are a sequence of genetic information that functions in a coordinated manner. It has several parts to it. It has an operator, a promoter, and two or more structural genes, right? So there's an operon, right, this operating system that controls the gene expression that we're going to talk about. So what is the job of the operator? The operator is a segment of DNA that the regulator binds to. So you have a segment of DNA, right? And a protein is going to bind to that segment. If that protein is an inducer, it can bind to the regulator in a way that interacts with the operator. If it decreases the effectiveness, it's referred to as a repressor, right? So if it's stopping something from happening by it binding, right, it's referred to as a repressor. If it increases the effectiveness, it's referred to as an activator. What we're dealing with in this system is a repressor. It actually stops the ability of the information of the DNA to be read by the organism. So sometimes it makes more sense when we look at the pictures. So don't fret just yet, right? The promoter is the region of the operon that acts as the initial binding site for RNA polymerase, right? So if you have DNA, what do you have to be able to do to make it into protein? You got to copy the DNA into what? Messenger RNA, right? And that messenger RNA then has to be read by the ribosome. In order for DNA to be made into RNA, the enzyme RNA polymerase must be able to bind to the DNA and copy it. If it can't bind and copy it, then you can't make that protein, right? So that region where there's actually a specific region where RNA polymerase binds to the DNA, that's called the promoter. That's not always accessible for RNA polymerase to bind to. And then, of course, RNA polymerase is the enzyme that actually binds to DNA, copies it into messenger RNA. Structural genes make something, typically a protein, right? That's the most common gene product, right? You've got DNA, you've got messenger RNA, you make a protein. One of the genes that we're dealing with today is GFP, which stands for green fluorescent protein. That gene, so that code in the DNA, made into messenger RNA, made then into that protein that when you shine UV light on it, it fluoresces green. Arabinose is a sugar. It's a 5-carbon sugar. It's actually from plants. And we're going to utilize that sugar today with this system. Normally, organisms will digest sugars, right? How do we digest it? What do we need to be able to digest a sugar? The enzymes, right? The enzymes to break down the sugar and utilize that sugar. So another very common operating system, right, an operon, is called the lac operon in E. coli. It allows it to digest, anyone know what sugar? Lactose. Do E. coli always see lactose? No. Most of the E. coli in our gut never sees lactose because you digest it in your small intestines and absorb it before it even gets to the large intestine. But your E. coli do know how to digest lactose. So if you become lactose intolerant, you stop, say, making enzymes to digest lactose, and lactose makes it to the large intestine, then E. coli is like, ooh, yum, right? I can eat lactose, but what is it going to have to do to be able to eat that lactose? Make the enzymes to digest it, right? Where's that information? In their DNA. Do they always use it? No, they don't always see lactose, right? So they have a way of sensing, oh, lactose is here. I need to make those enzymes that I know how to make in my DNA so I can digest this sugar. And man, they will do that lightning fast. They also need, remember, in order to take things into cells, sometimes you need proteins in your membrane, right? So they have some proteins that can detect the presence of lactose and bring it in. But when lactose is available, do you want to just bring in a little bit? No. You're going to make more of those proteins and bring in more lactose, right? I mean, as long as the buffet is there, eat up, right? And that's the way they look at it too. If it's there, they're going to bring it in. But they have to have some way, right, to sense that, okay, lactose is here. Let's make those enzymes. And this is true for most sugars for them, right? They're only going to make those enzymes if that sugar is available. So you have some control mechanism, right? That's why we have this operon, is to be able to control. When do we need to make these enzymes? Only when we need them. So what's a good inducer? What would be a good signal that says, okay, lactose is here, let's eat lactose? Why not the sugar itself, right? Lactose, right? If lactose is here, lactose is going to signal the system, okay, make enzymes so it can be digested. The same is true for arabinose. What happens with that sugar when it's present is it binds to the repressor. You see this protein, the ARC? See how this is actually a strip of DNA. It's binding to it, that protein. When it's bound, RNA polymerase can't bind to the promoter region right here and cannot copy the information on how to make those digestive enzymes. When the sugar binds, here, the repressor changes shape or maybe even detaches from the DNA. This allows the promoter to be available in RNA polymerase to bind, and so now it's going to copy this information. Right? It's going to make the enzymes to digest arabinose. So is that what we want our E. coli to do today? We're going to give it arabinose and have it digest it? Sure, why not, right? But instead we've tricked the system. Instead of the enzymes on how to make arabinose, we gave it the information, the gene, to make green fluorescent protein. So what happens is when arabinose binds to the regulator protein, the promoter becomes available, RNA polymerase binds, but now instead of making the enzymes to digest arabinose, it makes green fluorescent protein. We switched it out. Does that make sense to you guys? So when are they going to make green fluorescent protein? Only when what is present? Arabinose. Yep. Only when the sugar is present and it binds to the regulator is the promoter going to be available. RNA polymerase is going to bind, right? But instead of the normal operon where it's enzymes to digest the sugar, we stuck green fluorescent, or they stuck green fluorescent protein there instead. So if we don't give it the sugar, is it going to make green fluorescent protein? No, because normally, again, if you don't have a sugar, why would you make the enzymes to digest it? You wouldn't, would you? So the same is true for our bacteria. Unless we give it the sugar, it's not going to make green fluorescent protein because we put it in this operon for sugar digestion of arabinose. We switched it out.
Is the Arabinose enzyme gene removed or just disrupted?
The enzymes for digesting arabinose, I think they literally took them out and put green fluorescent protein there instead. None of those genes are there anymore in that operon. So we basically borrowed the repressor, how to make that repressor protein, right? The promoter site and the operator site where this is binding. We used all the regulatory functions of the arabinose operon, but instead of it having to make the enzymes to digest arabinose, we switched it out for green fluorescent protein. Does that make sense to you guys? So we genetically altered this operon so that in the presence, we could induce it, right? In the presence of sugar, it makes green fluorescent protein. Only in the presence of that sugar. So the regulator is the protein that binds to the DNA, right? And makes the promoter site inaccessible. The effector is a small organic molecule that binds, right? In this case, that's the arabinose that's going to bind. And it's going to make the promoter site accessible, right? RNA polymerase binds to the promoter, goes down and copies those structural genes. In the case of the pGlo system, it's going to make green fluorescent protein, right? The messenger RNA to make green fluorescent protein. In this case, the regulator is referred to as a repressor, right? It's stopping us from doing something when it's bound, right? The inducer is referred to as an activator because it's going to activate this system, right? When arabinose is present, it's going to activate the system. And of course, you got to make the messenger RNA to then make the protein, green fluorescent protein. So this is what they would normally do, right? They would take in arabinose, and this first gene product would help it convert it into this next sugar, and then the next enzyme into the next step in the process to break it down to be able to utilize that sugar. Right, so these are the actual structural genes that we've replaced for green fluorescent protein, right? Took them out, put green fluorescent protein in instead. We tricked it up. So green fluorescence is only going to happen with gene expression, right? Everyone agree with me on that? That's our hypothesis that hopefully we will prove that you're not going to get any glowing without gene expression. And if you don't get any gene expression, are you going to glow? No. Right? You have to express that gene, right? You have to make that protein in order to glow. So, two important genes, right? The BLA gene, this is to make that enzyme, beta-lactamase, right? To give them antibiotic resistance. Before transformation, does our E. coli have this gene? We're using it as a selective pressure, so it wouldn't do us any benefit if they already had it, right? So no. Before transformation, they do not have this gene. When we transform them, they're going to have it. We're going to give it to them. So the first step that we're going to do, right, is the tubes are already labeled for you in your bins. So go ahead and open your bin and you'll see two tubes, one labeled plus and one labeled minus, the bin, plastic, the big plastic bin. Right? So you see plus and minus tubes. Look at them and make sure they should already contain 250 microliters of transformation solution. So make sure that each tube looks like it has a little bit of liquid in them. So that first step has been done for you. You're going to get ice buckets out of the freezer. We're going to fill them with distilled water. Right? This is going to create a really cold ice water bath for our tubes. You're not going to use those foam racks. You're going to use those white plastic racks that you see that are floating racks. Although they won't float in our little ice buckets because they're too small, but they will float in our water bath when we go to the water bath in the back of the room that you see turned on with the lid open back there. Those foam racks that are in your cases are just for holding on to our tubes, and another tube that I'm going to give you later. They unfortunately stay wet if we use them for the other stuff. So we have our plastic racks for that. You're going to use a sterile loop, and you'll notice in your bin you have plastic loops. I'll show you how to take those out of the container. You're going to use those to take a little bit of bacteria from the plate that the little plate that you guys see that doesn't have the white label on it. It has bacteria growing on it. That's your starter E. coli. That's what it says, unfortunately, on that plate in my horrible handwriting. We've already divided up the solution, but one of the things we could have done, and I may do with my other class or next semester, is we could have added the bacteria to one of the tubes and then split it into the two tubes so that we get a nice uniform amount of bacteria between the two tubes. But in this case, we're just going to use the same loop because there's no difference between these tubes at this point. Pick up a little bit of bacteria and put it into each one of those tubes. And we do this on ice. Then I'm going to come around with the plasmid. I'm going to shine UV light on this plasmid. Why am I doing that?
Check if it works.
No? Because what is going to fluoresce if I shine UV light on it? What are the bacteria going to make? Green fluorescent protein, right? Are we giving them the protein, though? Today we're doing transformation. In transformation, what do we move? Genes. In the form of what molecule? A plasmid, which is what type of molecule? DNA. So if I have DNA in that tube and I shine UV light on it, is it going to fluoresce? It should not, right? Because the DNA is not what fluoresces. It's the protein that the bacteria will make. So if I've got glowing, I've got contamination with that protein, right? And we definitely don't want that. So I can tell you now, it shouldn't glow. If it glows, you'll see Miss Erica freak out. Someone's contaminated my plasmid with protein. So I'm gonna actually add, with a special pipetter, 10 microliters, right? I'm gonna use one of the micro pipetters into your plus tube, right? Because the plus is gonna be for the tube that we add the DNA to. Right. The loop is a pain in the butt, so I use the pipette mitt. What could you add to the negative tube to treat it as similarly as the plus tube? We're obviously not going to put DNA in it, but what else could we do? If we wanted a negative control, right? That's what this is. So my DNA is in a liquid, right? What's the most common liquid we deal with? Water, right? We could just add plain water, right? No plasmid as our control. We're not going to do that, right? It's not necessary to be that particular for this. So our negative tubes are going to be short a volume of 10 microliters. Woo. It's not the end of the world. Okay, so ampicillin, right, we already said this enzyme, right, beta-galactanase, that gene to make that enzyme, they don't have before transformation. We're going to give it to it. Also, when we transform these cells, are they going to have green fluorescent protein before we do this experiment? No, E. coli, none of your guys, if I was to grab E. coli out of the incubator, right, shine UV light on it, it's not going to fluoresce, right? Yeah, no. After transformation, will it? Yes. Will it always? No, what does it need to actually fluoresce? Actually make the protein? No. Arabinose, right? Because remember, this one is part of that Arabinose operon that we stole. So, we're going to leave the tubes in ice for 10 minutes. The plates have already been labeled, as you see, right? Each one of those plates is different. And because of that, it's easier for me to label them before I give them to you. Otherwise, you would potentially mix them up. Although our sugar is caramelized, so it turned the color of the plates a little darker. You're going to bring your whole ice bath, right? So the little buckets I give you with the ice, you're going to carry them back there to the water bath. And the reason why you're going to do this is because once you remove it from the ice and put it in the water bath, when you're done, you want to quickly move it right back to the ice. So it's easier if you just bring your whole bucket with you. And timing is important for this part of the experiment. You're going to transfer the rack, and so you'll also notice on the racks themselves, the white racks, there's a number, right? So that way you can remember that that's your group's rack, right? If all of you guys are back there at the same time, you know that that's your rack, right? So make sure that you take a look at that. Do not walk away. You're only going to put it in there for 50 seconds, okay? So time that, take it out, put it right back on your ice, and then transfer it back to your lab bench and it's going to sit on the ice for 2 minutes. So why are we doing this with our tubes? Why are we going ice to hot water to ice again? Yeah, it has to do with stress, actually. It's what we refer to as heat shock, okay, this process. And it's a stressor, and it's going to help get the DNA into the cells. So if you've ever gone on vacation, my hopes of this are dashing as we speak. It's okay. It's all good. Right? And you go where they have a hot tub and a pool. You ever jump back and forth between the hot tub and the pool? Right? It's kind of shocking to the system. That's literally what we're pretty much doing to our bacteria today, right? We're going pool, hot tub, back to the pool, right? We're going to shock the system. And we're using temperature to do that. It's really important, as I said, right? This has been worked out. 50 minutes is 50 seconds, not minutes. Oh, my Lord Jesus. Right? 50 seconds is the optimum, right? You go too long past that, your efficiency goes down, right? How many transformed cells you're going to end up with. So it's really important, right, that you don't walk away. 50 seconds goes by really fast, right? It's less than a minute. And I can say this from my experience in research where we had a little bit longer efficiency time, and I had a tendency to want to try and do something else, and then I would forget about it and have to start all over again. You don't want to do that. Right? 50 seconds. It's important. Okay, so 2 minutes. After the 2 minutes, then you're going to just take that white rack out and put it right on your bench top. The next step, I'm going to give you a little tube that contains 500 microliters of LB broth. It's food for your bacteria. You're going to take 250 microliters, and I'm going to give you the large pipetter and the large tips to do that. And you're going to transfer 250 microliters of this food from the tube I'm giving you into each one of your tubes. And as long as you don't touch down into the tube, right, you squirt from the top of the tube, you don't have to change your tips. But also maybe do your negative tube first, just in case, right? And then do your positive tube. So you don't contaminate positive and negative, right? So we aren't going to use these pipetters because you guys don't do so well with them, and neither do I, right? Because you would have to draw up to right here on that pipette, that plastic little pipette. You guys had trouble with 1 ml, never mind 250 microliters, right? So we're going to use the pipette men, which is much easier, right? And so I'll refresh you guys' memories on how to use that when we get to that step. Other classes, last night they used them. I noticed my stock is going down. So why were the vials, and we're going to, once we feed them, we're going to leave them for 10 minutes on our bench top before we put them on the plates, okay? So I want you guys to look at those plates for a second. What's in some of those plates? What's the difference between some of those plates? Look at those labels. What's in those plates? So LB is the food, right? What else is in some of those plates? The antibiotic what? Ampicillin. And one of your plates also has the sugar Arabinose, right? So y'all see that? So if we, and 3 out of those 4 plates contained ampicillin, yes? Okay. If we take the bacteria and we plate it right onto those plates without waiting the 10 minutes, will they be able to survive? No. Why not? Well, they've taken in the DNA, but what do they need to do with that DNA? They need to use that DNA to make what? The enzymes, right? So it's got to go DNA, messenger RNA, protein, right? Actually has to make the enzyme. Because if it doesn't make the beta-galactanase enzyme, what's going to happen? It's going to die when you put it on those plates, right? So ones that didn't take in the DNA, are they going to be able to survive on these plates? No, they're going to be killed, right? So the 10 minutes, right, is to give them the time to make, right, the beta-galactanase enzyme. That enzyme to destroy ampicillin so it won't kill them. So then, when we transfer to our plates, oh, you guys didn't take the little blue pipetters. That's okay. We'll get them for you. The little blue pipetters are like micropipetters, but they're fixed volume ones. So you don't have to worry about changing the knob. They do 50 microliters every time. But we want to plate 100 microliters on our plate. So what are we going to do? We're going to do it twice, right? You're going to squirt onto the plate twice, right? 50 times 50 is 100. Again, you could use those other pipettes, but again, doing that really small volume is even harder. So for the DNA positive tubes, notice you're going to put that on 2 plates, right? 2 plates say DNA positive. And then your negative tube, you're going to put onto 2 plates as well. You, of course, can use the same pipette tip for the plus tube for both the plus plates, right? But you need a different tip, or you can even use the other pipetter for doing your 2 minus plates from your minus tube, right? It's an important point. Do you guys understand what I'm trying to tell you? Okay. Okay. Plus tube onto 2 plates, minus tube onto 2 other plates. Don't cross-contaminate plus and minus. Right? Different tips for plus and minus. You don't have to do a different tip for each plate. Right? Just each tube. Okay. Use a sterile loop to spread out the bacteria on the plate. And again, one loop can be used for both plus plates. A different loop needs to be used for both minus plates. Yes? Okay. When you're done with your loops and your tips, they're going to need to go where? In the Lysol, right? Because they contain bacteria. Do not tape your plates together like it tells you in your lab manual, right? But do write your table number, your section. In this case, we'll write Wednesday, 8 a.m. and the date on all 4 plates. You can add to the label or with a Sharpie write on the side of the labels. Doesn't matter. So what information does those DNA minus plates give us? Why are we not adding DNA to one of the tubes and plating it? What is it for our experiment? It's our control. In this case, as it implies our negative control, right? It helps us verify that with the heat shock too, we didn't just kill everybody, that they can survive this process and they can grow on this media. And notice the media that we're going to put one of our DNA negatives on is not going to contain ampicillin. But the other plate that we're going to plate the DNA negative ones on does contain ampicillin. So these are our non-transformed E. coli, right? How they started out before the experiment. Are they going to grow on this ampicillin plate? Does E. coli naturally have resistance to ampicillin? No, we did this, right? In our Kirby-Bauer experiment, you saw ampicillin kills E. coli, right? It's a penicillin derivative that actually works against E. coli. So in this case, right, again, it's a negative control such that it should show that ampicillin kills E. coli. So this plate should have what? No growth on it. Still with me? All right, now let's move on to our plus plates. So transformation can occur only if the pGlo plasmid was introduced into the solution. Which plates are going to show transformation? These are our DNA plus plates, right? But we have 2 of them. as you guys already noticed, right, one of them contains the arabinose. Why are we using the arabinose? Why is it that the arabinose plate is going to fluoresce and the other plate, even though it has the DNA, is not going to fluoresce? Right. What's special about the arabinose? It's our activator, right? It's going to activate that operon, get them to actually produce green fluorescent protein. That's the only plate that's going to glow because it's the only one that has that activator.
Is it still considered transformation then with the ampicillin plates since they don't have
Yeah. So Alerica asked an excellent question. She said, this other plate, right, the DNA positive plate with ampicillin, are those still transformed cells that are growing on that plate? Yes, because what would happen to them if they weren't transformed? They die because the ampicillin selects for transformants, right? Having that ampicillin, only ones that took up the DNA and utilized it, at least for the ampicillin resistance, they won't grow in the presence of ampicillin. But as far as actually seeing green fluorescent protein, that only happens if they express the DNA. And remember, that DNA is under control of the operon utilizing arabinose. That's the important distinction between those two plates, right? They're both going to be transformants, right? But we're only going to see expression of green fluorescent protein with arabinose, with that activator. So as I said, the efficiency has been worked out already for this experiment. So we could technically count, although it's usually difficult because the plates we use are smaller because of the special reagents that I have to add. But this is very similar to what we did before with dilution, right? If you know how much volume you plated and you know how many cells you have, right, and how much DNA you added, we could figure out how efficient we are. So I'm going to skip over this for you guys today, right? I want to talk about it next time. So here are our 4 plates, right? So remember, this is our just the food, no ampicillin. We should get plenty of growth. Should show that our E. coli was viable to begin with. Our plate B is our DNA negative, right? Our non-transformed cells, our control. It contains ampicillin, so what should happen for this plate? There should be no growth. They should be killed. These are our 2 transformed plates, right? So only transformed cells, DNA positive, that actually utilized it and actually can grow now in the presence of ampicillin should grow. Will they glow, though? No, because this plate doesn't have what? Doesn't have the arabinose. This plate, on the other hand, right, has the arabinose, will therefore cause it to express the green fluorescent protein, actually make that protein actually fluoresce. So, hopefully your plates will, right, especially that one, will look like this next week. Right, and then you guys had the cool one I showed you already. You guys ready to do this?